I know, right? Thank you, everybody, for coming to Genetically Modified Crops, Benefits, Methods, and Concerns with Professor Patrick Byrne from CSU. I'm Andy Kennedy. I'm the Program Director of YVSC. It's a nice, intimate evening. Thought we'd maybe have a few more people, which is why we rolled out the preachers for tonight. This is really technically our second talk on GMOs, but over the course of the whole last year, there have been 4 or 5, maybe even more than that, I think 5. The library has done a couple talks on GMO, and our first talk was really kind of from the consumer side, with someone from natural grocers here, and someone that Georgie recommended to us that's been doing a lot of work on GMOs as well from the consumer side. And what we heard from the community was that we wanted to kind of display the other side and bring in someone that's really dedicated his life to the studies behind the crop genetic modification from CSU. So I'm going to let Todd introduce him, but before I do, a couple of little housekeeping things from YVSC. We have launched a really cool new program called 30 Friends of YVSC, and so you can take one of those little things to learn a little bit more about it, but we're trying to get 30 friends in 30 days, donating monthly to help us kind of spread out a donation, and as low as $5 a month. And at the end of the 30 days, which is July 8th, we're going to give away a Google tablet, which is a $200 piece of equipment there. So that's a pretty good odds. And then we're going to have a party at the Carnival Bar. The other thing that we have coming up is the Tour de Steamboat in July 19th. It benefits 4 local nonprofits, 3 different rides. So there's information about that over there too. There's a bike drawing. You can enter to win $7,500 Ericsson cycle, which is pretty cool. And a couple of other things on there, a wine drawing, and just a general live auction. So more stuff about YVSC on the table over there if you don't know about us. And let Todd take it away.
Thank you, Andy. We said it's going to take both of us to manage this crowd, so we'll tag team it here since there's so many here. Can you hear me in the back? Okay. We're using the microphone because Andy is recording it, so we hope this gets a lot of hits on the YVSC website. I'm Todd Higgins, the Agriculture Extension Agent here for Colorado State University. Extension is that branch of the university that's out here in the community. So what we do is we let the professors and the researchers on campus know what do we need to find out that helps the consumers and constituents out in the countryside. What research can we do that helps them out? And we partner with the university to conduct a lot of that research. What's interesting is my job as an agriculture extension agent is I get to see the whole gamut in the kind of GMO, organic, what do we use, herbicides and pesticides conversation. We've got some folks that have no problem using pesticides and herbicides, and then we have people who want to make sure that they're completely organic. And it's my job as the agriculture extension agent, and that's what we strive to do in our office, to find the balance, find what is the truth, what is the research behind why you should do one thing, why you should do another, and how we're going to feed all people, not only of our community, but of the world. I think we're looking at 9 billion people that we need to feed by 2050. That's a lot of folks. And there's room at the table for folks who want to do it all organic, and the people that need to do it more traditional ways. And that's what's great about having this conversation in Routt County. I wish there was more people here, because in Routt County we do a very good job of having conversations about wide-ranging topics in constructive ways. And I know that's what we're going to do here tonight with Dr. Byrne. I had the pleasure of hearing Dr. Byrne at an extension event in which he gave pretty much this presentation. And I said, finally, somebody that's kind of right here in the middle and I understand from a research perspective the benefits and also the concerns of GMOs. So I hope that's the light in which you're looking at this tonight. Dr. Byrne is a professor in the Department of Soil and Crop Sciences at CSU, Fort Collins, where he teaches and conducts research in plant breeding, genetics, and biotechnology. His research focuses on applying quantitative and molecular genetics to crop improvement, with an emphasis on drought tolerance and bread making, quality in wheat, disease resistance in dry beans, and seed yield in canola. He's been involved in a number of projects to evaluate the benefits and risks of genetically engineered crops. Prior to joining academia, Dr. Byrne spent 10 years with international agriculture development agencies in Nepal, the Cape Verde Islands, and Mexico. So please join me in welcoming my colleague, Dr. Patrick Byrne.
Well, good evening, everyone. It's my pleasure to be here. I want to thank the organizers for giving my wife and I the chance to have a beautiful drive over from Fort Collins, came up Poudre Canyon, and just green like we've never seen all the way here. So I hope you have a chance to do that trip sometime. Well, this is a topic that I find very interesting to talk about, and I hope that we'll be able to provide some useful information to you, but I hope it's a two-way conversation too. I want to know what's on your mind, what are your concerns. So, as you know, one of the things that characterizes this subject is opinions tend to be polarized. We hear very positive things about, for example, virus-resistant papayas that saved the industry in Hawaii. And we hear very fearful things and see lots of images like these. But to my way of thinking, the truth is somewhere in the middle. There's this huge gray area that reflects the fact that there are some benefits to the technology and there are some concerns. Maybe in some cases the use of the technology is justified, maybe in others it is best not to use it. So I hope to provide kind of a nuanced view, not yeah or no, but somewhere in between. So here's what I would like to do is to provide some introductory material, and then, you know, us professor types love to give quizzes, so we'll have a pop quiz. Tell you about GE crops on the market now, how they are produced, some potential applications, and then get into what are some of the concerns with the technology. So this is something, pardon me if this is just getting too simplistic, but just so we're on the same page. A gene is a segment of DNA that encodes a protein, and it does that by first being transcribed in the messenger RNA, translated into a protein, which then in some way gets expressed as a trait. And what makes genetic engineering possible is that virtually all forms of life use the same language. They use the same DNA alphabet. So it's very conceivable to take a gene from bacteria or another plant and put it into a crop plant. And that's really the motivation and the power of the technology, is that it allows one to draw upon the whole kingdom of life. So, what are genetically engineered plants? Indicates gene transfer using recombinant DNA technology. And what that means is a series of techniques for cloning, splicing together different segments, and then transferring that DNA into a cell or a tissue of another organism. Synonyms are genetically modified or genetically modified organism, transgenic, transformed. These all mean the same thing. I tend not to like GM or GMO, but it's perfectly okay if you use that. I've lost the battle because that's what's in the popular usage. I have found it necessary to include this, or at least informative, that hybrid technology is not the same as a GMO. Some GMOs are hybrid varieties, some are not. Hybrid technology is completely different. It's a technology developed in the 1930s based on crossing two unlike plants together, sexually compatible plants. Okay, the reason that I tend not to like the term GMO for this current technology is that we have been modifying plants genetically for thousands of years. And this is meant to represent the first farmers in the Middle East who were harvesting wild wheat and barley on the hillsides. And in the process of doing that, they were selecting seeds that were bigger, that were more adapted to that area, that had seeds that stayed on the plant. And the effect of that was to change the genetics of that population. So that is something we've been doing for a long time, not just something we started recently. Now, Mendel was the Austrian monk who worked on peas, and you all know this story. But when his results were rediscovered around 1900, that was really what set off the modern era of plant breeding, scientific plant breeding that's continued to this day. Okay, so here's the quiz part. So here's just a series of questions, and I would like you just to, by show of hands, tell me which one you think is true. So the statement here is fish antifreeze genes have been inserted into strawberries for cold tolerance. The idea is that these were genes from an Arctic flounder, which obviously is adapted to very cold temperatures. If you put that gene into strawberries, they would be able to resist cold and frost better. So the choices are that this is true, and they're currently on the market, true, and they're in the pipeline for commercialization, true but only experimentally, or false. This has never been reported. So how many of you think this is true and you can currently buy these? Okay. True, and they're in the pipeline. True, but only experimentally. And false, never reported. Okay, well, the answer is true. It was done. They exposed the strawberries to the cold. The strawberries turned to mush. It didn't work. End of project. But the image was too good. So the image lives on.
Why did it turn to mush?
Because the question is, why did the strawberries turn to mush? Because the protein was not effective in preventing ice crystal formation. Okay, this statement is that Bt cotton with resistance to lepidopteran pests has caused a major reduction in chemical insecticide use. And this is showing cotton. This is one of the crops where Bt genes have been inserted. So how many of you think this is true, that there's been a reduction in pesticide use? And how about false? Okay. It is true, and in fact, this is one of the major benefits of the technology, that the application of chemical insecticides has been reduced, and particularly in cotton, which really attracts a wide range of insects. Okay, Bt is also inserted into corn. The statement here is that widespread planting of Bt corn has led to field resistance in some of the target pests. How many of you think that that's a true statement? And false? Okay. It is true. It doesn't mean all target pests everywhere, but at least in some local populations, resistance has developed. Okay, if pollen from a transgenic crop cross-pollinates a conventional plant, the resulting seeds will be sterile. How many think that's true? And false? Okay. You are correct, the majority of you. But you would be forgiven if you thought elsewhere based on some of these images that show up on the web. In fact, the technology exists to do that, but it's never been commercialized, and the companies say they will never implement that technology. Okay, genetically engineered crops are regulated in the U.S. by, and then it's USDA, Environmental Protection Agency, or Food and Drug Administration. So on this one, you can vote more than once. So how many think USDA is involved? Okay, how about EPA and FDA? Okay, I think all of you are right at least once, because All of the above are involved in some aspect of regulation, but not in every case. And it depends on the legal authorization. USDA is involved in virtually every case where there's a field release of an experimental transgenic organism.
I'm sorry, can you go back to that last slide and explain what you mean by it's possible? About
This one.
yeah.
Okay, there is a molecular mechanism whereby a transgene, or a pollen from a transgenic organism will cross-pollinate, and then the seed that results will not be able to germinate the following year.
Okay, so then, but why is that false?
Okay, the technology exists, but has never been implemented. So it's false that transgenic crops, as they are currently implemented, that is not a true statement. Yep. Okay, so USDA involved in every case. EPA only when a pest or pesticide trait is involved. FDA only when food, feed, or pharmaceutical is involved. Okay. And this is the last question. How many different genetically engineered plant species are currently present, either fresh or processed, in U.S. food markets? And there's a range of those. So how many of you think it's in the range of 1 to 10? Okay, 11 to 50, 51 to 100, or more than 100. Okay, some of you voted for each of these, and talks I've given, this is the question that most people are guess wrong, or think differently than the true situation. It's really a very small number. The actual number is 8. Only 8 species. Now, some of them happen to be large acreage species, so the number of acres affected is large. Yes. Okay. Right. Right. And that's what the question is, how many different plant species are involved? So regardless, corn is in thousands of products. That's just one product. One species. Okay, so the follow-up question is, what are the 8? And you can just shout out your soy, corn. Sorry?
Wheat.
Nope, not wheat.
Alfalfa.
Alfalfa.
Canola.
Canola.
Sugar beet.
Sugar beet. Not rice. Pardon? Is not approved anywhere. Not in the U.S. Okay, I showed a picture on the first slide. Papaya. Cotton, I don't think you mentioned. And the other one is squash. Some forms of summer squash, but very small acreages. Okay, so there they are. Now, a larger number of species has gone through the regulatory process, has been approved, but they're not commercialized for whatever reason. Okay, here are the big 3, cotton, soybean, and corn. So since their introduction in '96, you can see that they've been adapted very rapidly by farmers, so that about 90% of the acres of those crops in the U.S. is genetically engineered. So regardless of what you think of the technology, it's impressive that a technology has been adapted so quickly. The single largest type of transgenic crop is Roundup Ready, or herbicide tolerant. And these are crops that have a gene that makes them resistant to the herbicide Roundup. Okay, Roundup is a general herbicide that will kill just about everything that's green, except plants that have a gene to resist the effects. So this is meant to show a weedy soybean field and then a field that has been sprayed with Roundup. with Roundup Ready soybeans. So farmers have liked this because it's a very effective and flexible weed control method. Just a single product is required. Reduced fuel and labor costs because fewer trips across the field. This is an important one. Facilitates no-till and reduced tillage because of its effects on weed control. And reduced tillage means soil conservation, water conservation, very important in a state like Colorado. And environmentally, it breaks down relatively quickly. So contrary to other products on the market, it does not leach into the groundwater. So environmentally speaking, glyphosate has some real advantages.
How can you say for sure that it doesn't leach into the groundwater? How can they say that?
Well, by testing, by taking samples of groundwater at different depths and seeing if Roundup or its breakdown products are there, and they don't. They stay pretty close to the soil surface.
Why?
Why? I think it's because the chemical properties that bind them to organic matter, which tends to be at the top on the surface. Right. Okay, so this does not mean that everything is rosy with Roundup Ready crops. The use of this technology on such large acreages means that resistance has developed. Now, it's very possible, depending on what part of the country you live in, that a farmer can have every crop in the rotation Roundup Ready. You could grow Roundup Ready soy, followed by Roundup Ready corn, followed by Roundup Ready cotton, followed by Roundup Ready alfalfa or sugar beets. And that's just a recipe for resistance to develop in some populations of weeds. So what this graph is showing is that globally in the U.S., the number of Roundup Ready, Roundup resistant weeds has increased a lot. And in some cases to the point where farmers can't use the Roundup Ready technology because of resistance. Now here, I love this picture. Maybe you know the weed kochia, which is one of those that produces tens of thousands of seeds on a plant, and it can break off and act as a tumbleweed. And this is a picture taken in eastern Colorado by one of my colleagues, and it's showing what happened when a kochia plant that developed resistance tumbled across the countryside, dropping its seed as it went. And you can see exactly where that plant went. And then the following year, the farmer sprayed Roundup, and there's the Roundup-resistant kochia. You can just imagine what happened the following season when each of these plants dropped seed. So it doesn't mean that this land is lost to agriculture, but it does mean that a different weed control strategy would need to be used. Okay, and then a different type of technology is drought tolerance. Very much on our minds in Colorado. Monsanto released a drought-tolerant corn in 2013. The gene involved is, interestingly, a cold shock protein that protects the RNA so that in normally stressed plants, gene expression would stop. But in these plants, because the RNA is protected, gene expression can continue. Now, I haven't seen the data, so I am not sure how effective this is. If it's only effective in a moderate drought or in a severe drought, I'm a little bit skeptical that a single gene can protect against all types of droughts, because drought is just so varied and complex. But at least in some situations, perhaps beneficial. Okay, so I would like now to get into producing transgenic plants, and it's a complex process everywhere from detecting the genes and isolating them, cloning the genes, putting together gene segments for a transgene, as I'll explain in a minute, transforming, and finally plant breeding to get the transformed gene into the plants that are actually commercially viable. And would you mind changing the battery here? There, maybe it's working. There it is. Okay. Okay, so this is a very simplified diagram just to show that genes inserted into plants with this technology have three parts. There's a promoter, which is like a light switch. It says how much of a particular gene product is made, where in the plant, when in the life cycle. So a very powerful controlling element. Usually a constitutive promoter is used, meaning it's turned on in most tissues most of the time. Then there's the coding sequence, that's for amino acids for the protein that you want to be expressed. And then a termination sequence, just saying this is the end of the gene. Okay, oftentimes these three segments would be from different species spliced together in such a way that they would be active in the plant tissue. Okay, then you have to get that segment into a plant, and there are two major ways of doing that. One is called a gene gun, and it actually works like a gun in that microscopic pellets of gold or tungsten are bombarded into plant tissue along with the DNA segment. And it seems very destructive, right? But in a certain number of cases, the pellet passes through the tissue and the segment stays behind and gets incorporated into a plant chromosome. So it has to be done many times with many different cells and tissues in order to get plants that actually show the effect of the genes without too many damaging properties. The preferred way of doing it is using a biological vector, something called Agrobacterium tumefaciens. And it's a great story I wish there was time to explain, but this bacterium in nature will transfer some of its DNA into a plant cell and cause a disease called ground gall. But what genetic engineers have done is to remove the disease-producing part of that gene and replace it with the gene of interest for the trait. So the gene transfer properties remain without the disease-causing properties. Okay, so this is all very brief, but after the gene is inserted, you need to grow the whole plant. If you can't grow an entire plant and get seeds back, then it's really not a viable project. And so this is usually done in tissue culture, and it's been a real stumbling block with some species, but usually with enough trial and error, whole plants have been regenerated. Okay, these are some future applications. Actually, this one is already released in Bangladesh. It's a BT, insect-resistant eggplant. We think of eggplant as an extremely minor crop here, even though I think it's a really delicious one. But in some parts of the world, it's a big part of the diet. Apparently, in South Asia, many applications of insecticide are required to get a harvestable product. So this would reduce the need for that many applications. Released in Bangladesh, not yet. In India, there's a court-ordered moratorium.
Is that put on by the Indian government?
Yes, that is. Right.
Because?
It's very political. It was approved by some branches of the government, but then not approved by other branches. So I think it's really a political decision, partly related to multinational company distrust of the multinationals. Okay, citrus greening disease has been described as the biggest threat ever to the citrus industry in the U.S. First detected in Florida in 2005, but affecting about a quarter of the crop now. Unfortunately, the recommended practice for controlling it is burning the diseased trees, not a very good control method. It has drastic effects on fruit size and quality, and it seems to defy traditional ways of controlling the problem. It is transmitted by a leaf hopper, and so one of the ways is multiple applications of pesticide, which is not something we tend to favor. So genetic engineering has been proposed. They have a gene from spinach, which is now in a 2 or 3 year-old tree. There are tests underway to see if this is going to be effective. Another example of a tree application is American chestnuts. Used to be the dominant tree in the eastern half of the country, but almost completely wiped out by fungal disease. Researchers in New York State have inserted a gene from wheat to control the spread of the fungus, and they reported recently that there is at least some degree of success in developing resistant trees. So maybe our grandchildren will know what a chestnut tree looks like. Biofortification. Two examples are cassava, which is grown in a very major part of the diet in some parts of the world, but extremely poor nutritionally. So a project in the U.S. was to enrich cassava for many different nutrients and other traits as well. Apparently, this has been moderately successful, and there are field trials in 2 African countries. And then, as you mentioned earlier, golden rice, which provides, at least the most recent version, of the amount that a normal person would eat would need 80% of the requirement of vitamin A. So considered important in rice-eating countries where vitamin A deficiency is common. Not yet released in any country. I understand the Philippines is the closest to approval and growing it. Okay, so we've been through how do you make them, what are the current applications, what are some of the future applications. So now let's get into some of the concerns, and I'm going to take a drink of water first. Okay, so one type of concern is ethical. Is GE technology playing God? And there's Michelangelo's view of God. You might think of God in different ways. But some people just feel that this is something that humans should not be doing. We shouldn't mess around with the genetic composition of another organism. I'm not going to talk about that because that's for a different venue. Another series of concerns are economic. Things like concentration of the seed business in the hands of a few corporations is risky. And I think it's a fair statement that biotechnology has driven consolidation. Consolidation probably would have happened anyway, but biotechnology just pushed it forward. Another economic concern is cross-pollination. Introduces GE material into organic food products, possibly reducing the value of the organic product. I'll show you some data about this. And then there's environmental and food safety, which are the 2 classes that I think science can provide some answers for. As far as environmental effects, this is a report that I like very much. This was done by the National Research Council of the U.S. I think it's a very balanced report. It was a large committee drawing on a diverse range of people that put it together. And I think they were very fair and honest and rigorous in their assessment. And they say, in general, the committee finds that genetic engineering technology has produced substantial net environmental and economic benefits to U.S. farmers compared with non-GE crops in conventional agriculture. Okay, so notice that, sorry about that. So notice that they say, in general, not always, everywhere, and they say compared with non-GE crops in conventional agriculture. So they're not comparing this to organic agriculture, for example. And then they go on to say, however, the benefits have not been universal. Some may decline over time. And so I think that's what's happening with the round-up ready technology. What was really a beneficial and powerful technology when it was introduced 15 years ago, the value of that is now reduced, at least in some areas. That same report, they also say the use of pesticides with toxicity to non-target organisms or with greater persistence in soil and waterways has typically been lower in GE fields than in non-GE, non-organic fields. And the adoption of herbicide-resistant crops complements conservation tillage practices, which reduces the adverse effects of tillage on soil and water quality. This may represent the largest single environmental benefit of GE crops. Okay. So I think based on this report, at least, there are some important contributions that GE crops can make. However, some concerns would be, what if these transgenes cross-pollinate and get into wild populations? Every crop that we grow has got wild relatives somewhere in the range. Now, this really would depend on which crop you're talking about and which location. Because, for example, there are no wild relatives of corn in the U.S., so that is not an issue here, but it would be for Mexico, where corn was domesticated and where there are still many wild relatives. For Colorado, what is of concern would be jointed goatgrass and wheat. Now, there are no GE varieties of wheat on the market now, but there may be in the future. And so this is some research my colleagues and I undertook in eastern Colorado to see what's the frequency with which winter wheat, shown here, cross-pollinates with jointed goatgrass to form these hybrids. Jointed goatgrass, maybe you know, it's an invasive species. It can get into wheat and really make farming wheat harder and decrease the price that farmers receive for their harvest. So we evaluated the frequency of these hybrids. Most of these hybrids are sterile, and so that would not be a risk if they all died. But not all are sterile. And so we evaluated what are the percentage which can cross back to jointed goatgrass, so that a transgene can get from the hybrid back into the wild population. We did this for several years, and the bottom line is that, yes, gene transfer happens, but it's at a very low percentage. The average was 0.1% that you have from this stage back to the jointed goatgrass. So certainly a concern, and I think if GE wheat was approved, that EPA would require that there would be stewardship to prevent something like this happening. A scenario that would be very negative is, what if a drought-tolerant gene got into jointed goatgrass and made jointed goatgrass even more invasive than it is now, and maybe it would get into rangelands, not just into cropland. So there are other concerns with similar crops, but this is the one of greatest concern in Colorado.
What did the kernels look like on that hybrid? Were they usable at all?
On that hybrid? No, they're not, because the seed is surrounded by a very tough seed case that you really need pliers to break into. So not a very good food crop at all. Okay, this is some interesting work that some colleagues and I did in Boulder County. There was concern by organic growers, what if GE, BT gene, for example, got into organic corn and decreased the value? How far would you need to separate GE from organic field in order to have the cross-pollination rate below a certain level? And so what we did was to plant an island of blue corn, with blue kernels corn, in the middle of a big field of yellow corn. Now, I felt great about this project, and this was probably the most fun I've ever had on a scientific project, because in a previous job, I had worked on the genetics of blue corn, and I knew that if you had a single pollen grain from a blue corn plant land on a silk of a yellow corn plant, that kernel would be blue. And so by planting this here and then sampling in each of these dots, each dot represents 10 ears, and we could count the number of blue kernels at each of those distances and have a very sensitive way of tracking pollen flow. No lab test required at all. So here's an example of our data. These were the two parents up here. Oh, and then you can see at 2.5 feet, this would be the first row of yellow corn adjacent to the blue corn. About 50% cross-pollination. Well, we know that's the way corn works. It's a wind-pollinated crop, and so we expect that. But then it drops off very quickly, so that by 30 feet, down to 14%, by 150 feet, less than 1%, and then a long tail out to 600 feet. So we plotted this here, and it was interesting that we found very repeatable results. We did this for 5 years in different locations in Boulder County and got very similar results every time. In that cross-pollination started very high, dropped off quickly, had an inflection point, and then a long tail. So that inflection point was predictable. The tail was not. Tail becomes almost like a random event, because out at that far end would be maybe just one single pollen grain got out there. Anyway, I highlight the 150 feet because that's what the county commissioners of Boulder County decided would be a reasonable separation distance to keep cross-pollination below 1%. Okay, so the county, on county-owned land, will provide that 150-foot buffer if a farmer indicates he wants to be protected against cross-pollination. Yes?
Do you keep any data on wind or temperature humidity in these trials?
We did.
You can't necessarily take Boulder County and extrapolate that out to anything else that's not Boulder County, right?
Well, you can by comparing the data, and some of our results, we also had trials in Burlington and near Fort Morgan in Colorado. And we had very similar results, and actually our results weren't that different from the eastern United States either. We did track direction of wind, and we found that there was some effect of prevailing winds, but the winds in Colorado can change so much, even within a day, that there wasn't a huge effect. We couldn't say that if you plant on the east side or the west side, that you would be guaranteed protection. Okay, so, a lot of times the work that we do as scientists gets published in some relatively obscure place in a journal, and maybe a few people would read it sometime. But this was, I guess, the case where the results from an experiment actually got implemented into public policy. And so it made me feel really good that we could provide some type of input like that.
Dr. Byrne,
Yes.
did the public believe it as well? Did they accept it?
They did. Well, they accepted the fact that, yes, that this data was valid, and that if you planted at 150 feet, you would get cross-pollination less than 1%. Not everyone agreed that less than 1% was sufficient. One question was, how far would you have to go to get 0%? And the answer has to be, as a scientist, you can't talk about zero, because there could be some random event, some strong wind that would take a pollen grain a mile or something. But you could say very low level.
Was there some basis which was adopted when you said 1%? But, some
Right.
statistical basis?
At the time, it was what the European European Union had decided was the threshold for labeling of GE products. Now, since then, they've reduced it to 0.9%, but this would still be valid for that level as well. Okay, health effects. Okay, and there's lots that could be said about this, but this will be pretty brief. There is no credible evidence that GE foods are poor nutritionally, have caused allergic reactions, or have more toxic effects than their conventional counterparts. This also is from the National Research Council. It's about 10 years old, but since that time, I think there has not been anything to make this less true than it was then. A number of other organizations, American Medical Association, World Health Organization, etc., have also confirmed or supported the safety of GE food. So let me say that this is the mainstream view of the medical establishment. You know, there are other views as well, but as far as most of those organizations that we think of as being scientifically credible, this is what they said. Allergenicity is one of those concerns. There's no evidence that an approved GE product has caused allergic reactions. Now, there have been experimental cases. There was a Brazil nut gene put into soybeans to increase the protein quality. It was found that when they tested in a group of allergic sensitive patients that the soybean did provoke an allergic reaction, but that was the end of the project. It was trashed back then. So it shows both that it's possible for a transferred gene to cause an allergy, but also that the system of testing and regulation is pretty good, and that it picked it up before it was released.
So this testing was done in response to some set of federal government regulations?
It was, right. And I think that's my next slide, which shows how it's done. But it was also done because Brazil nut itself is known to cause allergic reactions in some people. And so it was an obvious one that they needed to test. Okay, so the way it's done, comparing the DNA sequence and protein sequence, biochemical characteristics to known allergens, determine amount of protein in the food. A larger amount of the protein would have a higher chance of causing an allergic reaction. Evaluate heat stability. If a protein is heat stable, it's less likely to be broken down by cooking, and so would have a higher probability of causing a reaction, as well as evaluating digestibility under stimulated gastric conditions, which means a very low pH like the human gut, to see if protein is stable. So it's considered a pretty good predictor of allergenicity, but not perfect. What researchers would really like to have is an animal model, strain of mouse or dog or rat that would have similar immune system and digestive system properties as humans. Okay, labeling of GE foods. This may be on the ballot in Colorado in 2014. I think a group has received permission to collect signatures. This is a fact sheet that I wrote a few years ago. Andy, how are we doing for time?
We are at 6:30.
Okay.
So, yeah, so long as we leave, 15 minutes for questions. So
Yeah.
about 15 more minutes.
Yeah, well, I'm nearly done, so.
Perfect.
Okay. So just a few comments on labeling. And the idea here is that I think the petition that's being circulated is asking that all processed foods that contain a genetically engineered ingredient, that be stated on the label. It doesn't apply to animal products that have been fed GE food, doesn't apply to restaurant food, doesn't apply to beer, wine. There's a whole range of exclusions, but it would apply to things like cereals or cake mixes or many of the processed foods. So these are a list of arguments in favor of labeling, that consumers have a right to know what's in their food, especially concerning products for which health and environmental concerns have been raised. That labeling would allow consumers to identify and steer clear of food products that cause them problems. Surveys indicate that the majority of Americans support mandatory labeling, but I think most of these surveys have not included anything about the economics. What would be the cost of labeling? And that many countries and the European Union have some form of mandatory labeling. And the U.S. could follow their lead in handling the logistics of product separation. Okay. On the other hand, the anti-labeling arguments are that labels on GE food imply a warning about health effects. In fact, there is mandatory labeling already required by FDA in cases where an allergic, nutritional, or toxic effect of the GE product has been demonstrated. In those cases, a label is required. But the petition that's being circulated would be all GE products, regardless of whether health effects have been demonstrated or not. Okay, labeling of GE foods to fulfill the desires of some consumers would impose a cost on all consumers. This $400, I'm not sure how rigorously that was determined. While campaign was underway in California, a University of California economist came up with this estimate that it would be the average cost for a household. Consumers who want to buy non-GE food already have an option to purchase certified organic, or there's a new class of certification of non-GMO project. Maybe you've seen that on certain products. And this is a big one, that the food system infrastructure in this country, all the storage facilities, processing, transportation, cannot currently accommodate the need for segregation of GE and non-GE products. It's pretty mind-boggling to think of all of the small elevators in Colorado having to have separate silos and storage facilities for their GE corn and their non-GE corn, and processing facilities that have separate conveyor belts. So that's where most of that cost increase would come from, not the cost of a label for the ink, but on everything that would go into testing of GE material.
What agency would be responsible to oversee that?
FDA, Food and Drug Administration. Yes.
State initiative?
This is, yes, you're right. Okay. At the national level, it would be the FDA. The Colorado initiative would be, that's a good question, would it be the Department of Agriculture? Or is there another agency that deals with food? Department of Health?
Well, my question would be if it passed, I mean, everything crosses, I mean, I don't know how we might not be, how that could ever work. Yeah, I don't know.
Except if you notice, go to your grocery store and start looking. I looked in my fridge today. They're already labeling. There's no GMOs in this product. I've got it. I'm surprised at how many products are labeled that already.
Yeah, that puts an emphasis on the non-GMO, and that's a huge cost.
Yes.
Counterproductive for those people who want to be non-GMO.
Well, if it passed, but it would only take a couple states to pass something like that, it would have to go national because Kraft couldn't sell something in Colorado that wasn't labeled, they'd have to.
Well, in fact, it has been passed in Vermont. I think it will take effect in 2016. And a couple of other New England states have passed something that would take effect when a certain number of contiguous states pass similar things. So you would need 5 states in New England to all agree, for example.
What happened in California?
California, Oregon, and Washington, it failed in all cases.
That wasn't to vote, though, that was legislatively.
No, that was a popular vote. It was in response to a petition similar to the one being circulated here.
How equivalent was the funding?
Pardon?
How equivalent was the funding spending on that initiative?
Well, I don't know. I don't know. Yes?
Who's to say that all products need to be labeled non-GMO, but maybe they all need to be labeled GMO, and then whatever person really doesn't want GMO can go to kind of a boutique industry.
Mm-hmm. Well, and that's, I guess, the situation we have now.
No, just like organic.
Yeah,
You
right.
pay a premium if you want organic product. Well, it could be that you want a premium product if you want a non-GMO oriented.
Right.
And
Well,
why would that cost be greater than for the general public?
yeah, I'm sorry I didn't follow the last part of the
It's still
question.
going to cost, because in order to label, it has to separate and process separately anyway, so the cost is still going to be there whether or not they choose to label. So the labeling doesn't end up being the
Well,
cost of the process.
But most people will opt, if most people opt to just go ahead and get their product, then it doesn't really matter. If I go to buy, let's say, Holly sugar, that's the first thing that came to my mind, and I wanted to get Holly sugar, I could choose to do that, and I could say on there, genetically modified sugar. And most people would prefer to do that than to pay the extra money it would take to get Holly special organic, non-GMO sugar.
Yeah, and I guess part of it is having that label on a product. Does that imply some health warning, or is it just a neutral statement that you can choose to pay attention to or not? And in the case of sugar, it's a good point because sugar is so highly refined. There is no DNA, there is no protein in that product. Is it right to say this is a GMO product because it was produced from a GM sugar cane?
You can also label it fat-free, a bag of sugar.
You could, and I think FDA doesn't like that type of labeling because it implies that some other types of sugar do have fat in them.
Not much different than an all-natural label.
Well, right, and all natural is completely meaningless, right?
Right, but nobody's educated about that, so that's the determination of organizers saying that they don't want these labels because it implies something that's not,
Mhm.
and not the public does their education.
Right. Now, I got an interesting phone call from Senator Udall's office a few months ago, and he was interested in doing something about labeling. He was saying that he would like to have a label connected to some kind of QR code or website where people can actually go to see what does this mean. Not just GMO product, but what is the source of the gene, what is the data that exists, just to provide a little more background information on the product. I thought that was an interesting approach. I don't think it's gotten very far.
What's that going to do to the small cottage industry? I mean, they can't afford to do that kind of stuff, to QR codes and sourcing all of their ingredients. I just don't know.
Yeah. Well,
Between the industry, basically, as it has been labeled as GMO, people with bigger industries, they can take the hit, they can do the packaging, they can do marketing. Because the assumption is that the food you're buying is not altered in any way. Then the little growers, the organic growers don't have that burden. And
But I think with the
so-
local board interest that, that should push that.
But the organic grower, that organic label already implies non-GMO, because that's part of the definition of organic certification. So that's already embodied within an organic label.
And they pay a premium for that.
Right, and they charge a premium as well for that.
Without processing is correct.
Right.
Full disclosure, without processing is correct. Organic growers don't enjoy the same processes that their conventional counterparts do, correct?
Well, I don't know. It depends on the crop. There's really just a small number of crops that
know
have.
the answer to that.
Well, there's only a small number of crops that receive subsidies, right? And I believe if a farmer qualifies for a subsidy for wheat or corn or cotton, it doesn't matter if it's organic or not. But why don't you let me finish this, and then we can have a more general discussion. So, I think this is the last slide. Just some of the important questions, in my mind at least. What's the most effective strategy to minimize the development of insect and weed resistance in response to GE crops? How can we get better stewardship processes on behalf of the farmers and the companies so that we don't have something like this Roundup resistance continuing to spread? What's the environmental impact of gene flow from GE crops to related wild species? What's the appropriate level of regulation that protects health and the environment while rewarding innovation and improving food production? Okay, so I think this is an important one that a number of the comments relate to. How much regulation do we need? And how are we going to improve food production, not just in this country, but around the world? And can GE technology be a part of that? You know, certainly not the complete answer, not the silver bullet by any means, but can it be part of that? And what are the best ways to ensure coexistence between GE, conventional, and organic crops? I think all of us would like to see these various types of systems continue and have farmers have the option to choose the system that they want to grow. But can they coexist together, or is coexistence impossible? I guess I like to believe that coexistence is possible with spirit of compromise and a willingness to work together. And at least in Boulder County, it was really gratifying that neighbors, when they got to know each other and talk about their situation, they were willing to compromise and reach a solution. So I would like to believe that that's possible. So, I've got a few references there, or if you're interested, and thank you very much for your attention, and look forward to continued discussion with you. So, I think you have a question.
See how I can formulate it. So I would like you, if you could, to discuss the rate of development of the BT gene splicing process, and what its intentions are, and how it, what are the symptoms of the pest that eats the BT products?
Okay. The genes are from the soil-dwelling bacterium, Bacillus thuringiensis. They're called crystalline genes or cry-genes because they form a crystalline protein that in the insect gut it dissolves and binds to a receptor on the gut lining. Now the insect gut has a pH of about 6 to 8 compared to mammalian guts, which are 1 or 2. And so these toxins do not dissolve in a mammalian gut and have no effect at all. But it's really the specificity between the binding of this crystalline protein and the receptor on the insect gut, which causes a pore, which causes massive influx of water into or loss of water from the digestive system. And it effectively paralyzes the digestive digestive system, so that after a couple of days, the insect starts to work.
I have a related question about BT, because I know having used it for insecticide in gardens, there's all kinds of warning labels for just handling stuff. So if it's in the food you're eating, there's got to be some issues there, even though you're saying pH is a different level for insects to humans. So have there been any studies on the persistence of BT in food and the effects of that in human beings over time? Because we've only seen this in food since 1996 maybe. There can't be any seriously long-term studies that have even been done, correct?
Well, I guess it depends on what long-term means. Certainly in laboratory animals and livestock, that there have been tests done, and fragments of genes and proteins are found, but not intact protein.
What do you think the effects of the BT gene would be on pollinators, which is obviously the most important piece of the whole agriculture system?
Right. There are different versions of BT genes, and some of them target particular classes of insects. So there are some that would target the order that bees and other pollinators belong to. The ones that are implemented in crops are targeted to Coleoptera, meaning beetles, and Lepidoptera, moths and butterflies. Part of what EPA requires is tests on non-target insects, and maybe you heard about the story with the monarch butterfly, that there were some lab tests showing that BT pollen caused higher mortality rates in monarch butterfly compared to non-BT pollen. That was done in a lab. When it was repeated in the field over many years, it was shown that there was no effect really on monarch population. So pollinators is something that is incorporated in the testing and evaluation of these crops. I guess I feel that one of the shortcomings of our regulatory system is that it doesn't include a post-commercialization monitoring, or at least that monitoring should be more rigorous to show, okay, if something didn't show up in the pre-release, maybe it would be detected after things were grown on a broader scale. Okay, that's done to some extent, but it seems to me it could be done more broadly than it is. Yes?
The whole concept of the genetic engineering that's taking place now by a relatively few companies. What did you say? Well, how many mega agricultural companies are doing this? Half a dozen? Something?
Yeah, 6 or 8.
Something like that. What it represents, on the one hand, is even further simplification of the gene pool, because they're spending enormous amounts of money trying to develop these few strains of very expensive plants. On the other spectrum, there's the whole theory of resilience and sustainability of systems. And one of the basic tenets of resilience theory is that with simplification, you are going to run a real risk of losing resilience and ultimately sustainability. So how much attention in this march toward these products, how much attention is being paid to these broader concepts of maintaining diversity in the gene pool?
Yeah, that's a great question. And genetic diversity is on the minds of a lot of people. I should say that when a single gene like a BT gene for European corn borer is developed, it's not just a single strand. It's put into a whole range of different corn hybrids adapted to different latitudes and temperatures and length of growing season. And so it may not be as bad as what you're thinking. The companies are not dumb. They realize that for their long-term interests, they need to maintain genetic diversity. And so almost all of them have got programs, for example, where they're looking at tropical and subtropical strains of corn and trying to incorporate that into their gene pool. And certainly in the public sector, there's lots of work. I'm working to develop drought-tolerant wheat based on wild wheat from the Middle East. So there's a lot of that research going on. I agree with you. I think that companies could be doing more and paying more attention to that, because they're after the most elite, highest yielding, the thoroughbred racehorses, and need to be looking more at that diverse gene pool. Yes.
Just following on, how different or how big, how much larger a threat is genetic engineering approach to losing that diversity than traditional plant breeding for very narrow characteristics that benefit agriculture? Is that significantly higher risk with genetic engineering, or do we not have the same problem?
It's a similar risk that you would have with conventional breeding. And the example in conventional breeding is southern corn leaf blight, which decreased the U.S. corn yield by about 15% in 1970, wiped out a lot of corn fields. And that was well before genetic engineering.
And how do we address that in conventional plant breeding?
Right. Well, that event in 1970 was kind of a wake-up call, and companies started devoting more resources to a broader germplasm base. But like I said, I think they're not doing as much as they could. Genetic engineering is maybe heightens the concern, because those small number of genes are put into many different hybrids. But it's still, if there's something about that gene which would make the crop vulnerable, then that would affect a lot of acres. Yes.
How do you address the question that all of the research is being funded by Monsanto and the other big companies?
Well, first of all, that's not completely true, because USDA has a program called Biotechnology Risk Assessment. And so they are funding research in the private sector about environmental risks. I guess the research is done by the private companies, but then reviewed by the federal agencies. So FDA, for example, would look at all the compositional and feeding studies and say, okay, we accept that this is enough and you've done this well and we agree with your statistical procedures, or we think you need more, go back and collect more data. So I guess we need to depend on the integrity of the regulatory agencies to do a good surveillance and monitoring.
You suggest they have integrity.
Pardon?
You suggest they have integrity. So where's all the door?
Well, in some cases, it's not completely that way. I think there are many good ethical scientists of high integrity in the regulatory agencies and trying to do the best job. So I guess I feel that the current system of review is okay. It would be better if we had some third-party independent test as well, but that's not part of the system we have right now.
So what is plant selection, not genetic modification? If you do plant selection, old-fashioned plant selection, is genetic modification, or is that different?
Well, it's modification with a small m. It's changing the genetics by selecting certain plants and discarding others. It is more broad-based. It's based on selecting the effects of many genes together, many genes having small effect, whereas capital GM is having one gene with a very big effect. So that's one way of distinguishing.
Question.
Yes.
To the gentleman's point on genetic diversity earlier, I have to admit what I find so appallingly disingenuous about the proponents of genetically engineered papaya, for example. Having worked on fungal disease in the tropics, I can tell you from first-hand experience that fungal disease will exploit a monocrop, and I didn't hear anything about monocrops anywhere in the presentation. Genetic engineering is a system for breeding life support into dying monocultures, a system of agriculture that has proven to be a failure over the course of history and will continue to prove to be a failure as we go forward. Cavendish banana in the tropics will be extinct in 10 years. I worked on Diabroma cacao, and we had great success with horticultural methods, with other cultural methods, and simple, non-invasive, organic, sustainable, biologically diverse cultural methods. So to make a presentation like this and never once mention the word monocrop, I'm flabbergasted. I'm utterly flabbergasted.
Yeah, well, I think it's really two different issues. And I think
How so?
I think that you could certainly incorporate genetic engineering within a robust system of crop rotation.
It's irrelevant in a culturally diverse system. It's absolutely irrelevant because those ancestors 10,000 years ago were selecting for diversity. They were not selecting for a single gene type. They were not narrowing down everything into having a ET trait in their seed stuff. That is disingenuous.
Yeah, I don't know. I do believe genetic engineering could be part of an agronomically responsible, diverse
It's not
system.
necessary. It's not necessary. Where there is diversity, it's absolutely not necessary.
I'm sorry, I don't find them mutually exclusive. I think the stewardship of the farmers needs to be better than it has been as far as rotating herbicides, as far as rotating the crops, rotating tillage methods, all of which are going to give us a more robust
We're
system.
talking about monoculture, correct?
Well,
Rather than biologically diverse, organic.
well, monoculture in one season doesn't mean monoculture over a period of years where you have rotation of crops.
Well, then let's talk about papaya.
Actually, I'm gonna halt you right there, Antonio. We are at 7 o'clock, and just to respect everybody's time tonight, we can take maybe a few questions, but I know that maybe gonna take Patrick out to dinner. Okay. Don't want to make that assumption, but I guess I just did. So a couple of points I want to make before maybe we take some of these conversations offline is that Ron Kroll from Off the Beaten Path did bring a couple of books, Tomorrow's Table, Organic Farming, Genetics, and the Future of Food, if you guys are interested in that. And Ron is also a wealth of information on the topic, so he might be able to answer some of your questions too. Tonight was brought to you by, I have to mention our sponsors, the New Power Fund, Homeland Magazine, and of course the lodging sponsor, Mountain Resort. Thank you for the YVSC, and I appreciate everybody coming. So, if you have any other questions, we can take some of those conversations offline, but we need to close out tonight. So, thanks everybody. Thank you.
Yeah, I'd like to have a question.